my name is Ruth Conlin, and I'm from Drake University in Iowa. And my summer research is about linking genotype-specific inter-kingdom communication with shifts in environmentally relevant phosphorus pools, revealing new insights into plant microbe environment resource utilization. And so phosphorus use efficiency in agriculture is around 46% globally, which is not that great, and we'd like to improve that. And we know that the organisms in the rhizome microbiome are important in the phosphorus cycling and availability to plants, and polyphosphate accumulating organisms. They accumulate phosphate as polyphosphate, and they're really important in the phosphorus cycling and availability to plants. And so that's what my project is studying, the polyphosphate accumulating organisms, or PAOs, and their role in the different phosphorus pools available to plants. And so we grew different genotypes of maize under high and low nitrogen conditions, and then sampled the rhizome microbiome. And we sorted out the polyphosphate accumulating organisms using phosphate-activated cell sorting, and then we sequenced them to get the community structure. And then we also sequentially extract the different phosphorus pools that are relevant to how plants can uptake them. And then we use molybdenum to find out how much phosphorus is present in those different pools, and then we could relate the PAO community structure with the phosphorus pools. And so our results, we found that the corn genotype and nitrogen condition influences the PAO community, which is what we would expect. And then we also found that the growth stages and the genotypes influence the phosphorus pools, which is again what we would expect. And we found like a tighter clustering with the NRT1 genotype, suggesting that the loss of that gene is important in the phosphorus regulation. And then here, this is the PAO alpha diversity and the enzyme available phosphorus pool. And so we found a really interesting relationship between those two, like the sinusoidal relationship in both the wild type and the NRT1 knockout. So that was really interesting relationship to find. And then we also found that some relationships were different between the wild type and the NRT1 knockout. So that the NRT1 is also important in the phosphorus regulation as well as the nitrogen regulation. And then we found specific correlation between specific nutrient-relevant PAOs and the different phosphorus pools. Like nitrosphere is involved in the nitrogen cycle, but then it also has some correlations with the phosphorus pools, and methylobacterium is involved in the carbon cycle, but then it also has some significant relationships with the phosphorus pools. And future work can look more at the phenotype of the PAOs and their metabolism, what they're doing, instead of just the genotypes.
Cool. You did a great job. I'm just wondering, what was kind of like the biggest challenges you faced with this research project?
Yeah, the biggest challenge for me was just like learning how to do everything and learning how to like analyze the data some was really, a lot of learning for me.
Well, it looks like really nice analysis, so great job.